we start the next chapter in which we're going to talk about the microbial metabolism and genetics first, metabolism second. I will be focusing most in genetics. I will be focusing almost entirely on the basics of bacterial genetics. Okay, we're not going to talk about molecular biology and genetics of eukaryotes extensively because it's a nightmare. We can explain basics with bacteria, and that's the main, most relevant clinical type of pathogen. Same goes for metabolism. So it looks like it's smaller than other parts of the course. It's only 45 slides in the entire PowerPoint. But in terms of the concepts that are introduced and the concepts that we discuss, this is maybe the most the hardest, I would say. I refer to it's hardest because it's sort of a little abstract. You can't touch it. You can't really kind of see it in the regular life. It's a lot of chemistry involved. So stay focused, and it's doable, but stay focused, okay, when we talk about this. Now, a lot of the themes that we're going to touch on, you've already learned in other courses, in other biology courses, but mostly if you took any type of basic cellular biology, biology 1200 here or 1510, you already know a lot of things that I'm going to talk about. But it's never too bad to reiterate. So what we're going to start with is structure of DNA and RNA. We're going to start with the DNA, and one thing that we have to understand is that as any other biological macromolecule, DNA is a polymer that consists of building blocks. There are four different building blocks in the DNA: cytosine, thymine, adenine, and guanine. Okay, well, slightly different anyway. So it's C, T, A, and G, right? Now, individual building blocks of the DNA, bless you, are called nucleotides. Deoxyribonucleotides. Let's take a look at the word in more detail. So deoxy stands for the hydrogen atom in the 2' position. It means deoxy means there's no oxygen there, only hydrogen. Ribo stands for the sugar. The sugar itself, sugar without oxygen in the 2' position, is called deoxyribose. Okay? When deoxyribose sugar forms the bond, the covalent bond, with one of the nitrogenous bases, these two these two components of the DNA form the structural unit that is called a nucleoside. So I highlight it with the red square now. This is a nucleoside. Okay. When nucleoside gets phosphate residue added to the 5' carbon of the deoxyribose sugar, this whole thing is now called nucleotide. So when you see something like this DNA molecule has 300 nucleotides length, you know what it means. It means that the entire DNA molecule that we talk about consists of 300 individual building blocks, individual nucleotides. Is that clear? So understand why it is deoxyribose, what's nucleoside, what's nucleotide. What are the chemical components? Of course, standard question. do we have to draw deoxyribose? Do we have to draw phosphate? Do we have to draw all four nitrogenous bases? The answer is no. You don't have to like know the exact chemical structures, okay? But I will ask you, what are three components of a nucleotide? And you have to give me the answer. You have to list them. Not drawing them, and giving me chemical properties, no. Does that make sense? I will not ask you to recognize which nitrogenous base is this. Does that make sense? I will not give you a picture of this and say what it is. Right? Now, nitrogenous bases, there's four different organic molecules, belong to two groups. One group is called pyrimidines, right here, and another group is called purines. Just a chemical classes, sort of. And you can see the similarities in structure, right? This is the pyrimidine ring, and this is the purine structure. Now, if you want to know about evolutionary importance, why this? I will tell you because we don't really know. That's just so it happened. Okay. What is important to know is that this nitrogenous bases, they are parts of nucleotides that actually enable the formation of double helix, as we will see. Okay, they pair. You can think of them as having that important functional message. Okay. They carry the information, the bonds, the bases, okay? Now, what is the role for phosphate and sugar? Phosphate and sugar form so-called, surprise, sugar-phosphate backbone. Okay, so this is the backbone. You can see that two adjacent molecules of deoxyribose are connected by the phosphate. Okay? Does that make sense? Two ribose residues and phosphate in between them. Phosphate is between every two deoxyribose residues. And extremely important feature of that connection is that phosphate links 3' and 5' carbons. Often there's a question, why prime? Well, the answer is as simple as it is, in my opinion, kind of funny. Numbers without prime, without that little apostrophe in the top right, those numbers are assigned to carbons in the nitrogenous bases. To differentiate carbons in the nitrogenous bases and carbons in the deoxyribose, the ones in deoxyribose are numbered 1, 2, 3, 4, 5'. Okay. Now, what it means, 3' to 5'. So, if you look at this backbone, the sugar phosphate backbone, you will see that it's always 3' to 5', 3' to 5'. That makes sense so far? That they connected 3' to 5'. Now, the analogy that I can give you, I don't know the name of that dance, but you know, like when people stand one behind another and form that long line and then start to do like stupid moves with their hips and move forward, okay? They stand, What? Okay, a conga line. Terrific, great. So the idea is that I'm gonna face the back of the person in front of me, right? And every person in the line will face the back of the person in front of her or him. Nobody's going to dance standing back to back. It's very inconvenient, right? So the line has a certain orientation. Same goes for DNA. DNA has orientation. 5 prime, 3 prime. 5 prime, 3 prime. Look at this. You have 3 prime carbon of, let's name it deoxyribose 1, connected to 5 prime carbon of deoxyribose 2. 3 prime carbon of deoxyribose 2 connected to 5 prime carbon deoxyribose 3. So it's always in the same way, connection in the same way. That means that DNA molecule is directional. We can read it. We can read it from 5 prime to 3 prime. We can read it from 3 prime to 5 prime. Nobody bans us from doing that. It turns out, though, that in the nature, in the living nature, in the living cell, all enzymes that read DNA, enzymes that copy it, enzymes that make RNA on the DNA matrix, they all read from 5 prime to 3 prime. Does that make sense? Think about this. When you read the text, is it directional? Yeah, you read from left to right. Any clue why from left to right? Yeah, honestly, I have no idea why left to right, because in Arabic or Hebrew, it's right to left. We just agreed to do that this way, okay? We agreed to read from left to right. Does that make sense? Not to me, honestly. But okay, so be it. Left to right. For some reason, nature decided to read from 5 prime to 3 prime. And so it goes. Okay? So when we describe DNA molecule, we usually picture it like, say, I'm telling you, this is the gene, the fragment of DNA. By default, the scientific community agreed on placing 5 prime on the left and 3 prime on the right. It's just an agreement. There's no deep underlying concept. Just let's do it because we kind of, English is the language of science. It's all going to be written in Latin. Latin letters, so left to right. Does that make sense? Please understand the idea of directionality. That DNA is directional. Now, that's kind of a mind-blowing picture. So DNA is the helix, okay? Right-handed helix, double helix. I think I mentioned it. Yeah. April 1st, 1953, when Watson, Crick, and Maurice Wilkins published the paper on structure of the DNA, Nature. You can find it's freely accessible. It's like a page and a half. Paper that shows probably the highest, the biggest achievement of science. Well, I'm biased, of course, but I think it's one of the greatest achievements in science in 20th century. So every turn of the DNA molecule is about 10 nucleotides. There are multiple numbers. The distance between nucleotides, what the distance that one turn takes. I don't really care about the numbers, and I suggest you don't really care about the numbers, because if you're gonna study DNA structure and become structural biologist, it's a different story. You will memorize them. Otherwise, it's not that important. Okay. Now this DNA helix, what is interesting, remember we talked about directionality? Turns out DNA strands are anti-parallel, which means if one strand here has, let's assume, 5' to 3' direction, then another will be directed in the opposite way. Does that make sense? Like lanes on the road. Okay? They go in the opposite way. So that connection, that linkage, goes in the opposite way. And the fact that DNA molecule is directional and the RNA molecule is directional, it actually has great implications for the cellular processes, such as DNA transcription or DNA replication, that we're gonna discuss. Okay. Now, if you would look at the helix, we don't have a model in here, but even looking at the picture, you can see that there are two grooves, the major groove and the minor groove. These grooves are the sites for protein binding. DNA is the place for multiple proteins to interact. Histones, nucleoid-associated proteins in bacteria, DNA polymerase, RNA polymerase, a bunch of DNA binding proteins, when they interact with the DNA, they bind into the groove. That make sense? So these grooves are the sites for the binding, just proteins structurally fit in there. That make sense? Does it? Okay. Now, pairing. I don't know. How many times before you've heard about complementarity in the DNA? Like 5, 6, 7, I don't know, many, right? So I assume you've heard it. I just remind you that we have two types of pairs, and that's strict. In the DNA, the double strand is formed because A pairs with T and G pairs with C. Okay? So you have AT pair illustrated here and GC pair illustrated here. Okay? Now you can see kind of flattened out DNA structure on this illustration. Now imagine if you would try to make it in a helix. It's going to blow your mind. So you have AT pair and you have GC pair. What is the difference between them? The fundamental difference. Which one is stronger? Look, look, look. GC has more bonds. Which bonds? How do we call those bonds? Hydrogen bonds. Very good. You see, you have hydrogen bonds. Okay, so these dashed lines are hydrogen bonds. GC pair forms 3 hydrogen bonds. AT pair forms 2 hydrogen bonds. Why focus so much time on GC and AT? Now, look, we know in very many cases, we know the structure of the sequence of the DNA of some organism. And we can look at the sequence and say, we see the fragment of the sequence, and this fragment has a lot of A's and T's. From the structural point, what would it mean? What do you think? Easier to be broken down. Now, if you break down those hydrogen bonds, what's going to happen to the strands? They're going to get separated. Excellent. You see? So you break down hydrogen bonds, they get separated. Right? Now, based on that, you can make speculations about the function. For example, if you want to do the replication of the DNA, if you want to replicate the copy DNA, you have to separate the strands first. So it's likely that replication will start from the fragment which has a lot of ATs, so it's easier to separate initially. Make sense? Now, another thing. GC. They stable, right? Now, what can break down hydrogen bonds? What's the first thing that we have to think about? Temperature, okay? So higher temperature will break down the bonds. If you boil it, all bonds will be broken. Does that make sense? Now, there are microbes that live in very hot environments, physically hot, like 60, 70 degrees Celsius. Which pairs are going to dominate in the DNA? GC, of course, to keep DNA double-stranded. And you can actually see it if you sequence those thermophiles. They will have a lot of GC pairs, much more. Okay. Now the ratio of GC versus AT is called the Chargaff rule, or Chagraff. I always mix up G and R in his last name. So Chargaff rule says that in the DNA of an organism, the percentage of A's equals to the percentage of T. And same goes for G and C. Does that make sense? So if you have 20% of A's, you're going to have 20% of T's. Right? It doesn't mean that G, GC, and AT are going to have the same share. You're going to have different share. Okay? But Chargaff rule works pretty well. Since we talked about those bonds in the DNA, the process when two strands of the DNA get separated is called denaturation. As we just discussed, you can heat it up, right? You can heat up the DNA molecule and strands get separated. When they are separated, the bases become exposed. Okay? When they become exposed, they can be recognized by various proteins that can then read the information, because essentially those four letters, A, G, C, and T, give you a language. And those enzymes can read the information written in the DNA and can copy it or can express it. The one that we discussed, the cookbook to the dishes in the kitchen. Okay, does that make sense? Now, if you would look back at the double, well, actually, we don't have to go back. Look at the double strand. You see these guys, when they are in a double-stranded form, they're kind of hidden inside of the helix. They're not accessible. They make pairs, and they make like steps of the ladder. So in order to expose them to the enzyme, we have to separate the strands. Does that make sense? The strands are going to split. Now it's interesting, the process of DNA being denatured, the scientific jargon name for it is DNA melting. Of course, we do not talk about, DNA going from solid state to liquid state. We don't even work with the DNA per se. We work with the water solutions of DNA. That makes sense? Why melting? So DNA can absorb UV light at 260 nanometers, but that's not a point. Okay, it can absorb UV light. So the more DNA you have in the solution, the more absorption you're going to see. Make sense to you? Let's say you have a perfect solution of DNA in water, and that's absorption, and this is time. Okay? And you start to heat it up. While the solution warms up, nothing happens to the DNA molecule. It remains double-stranded. But at some point, at certain temperature, strands go into separate. Turns out, the parts of the molecule that actually absorb light are the nitrogenous bases. So when you get those bases exposed, the absorption increases several-fold. And what you're going to see is this. Okay? So this point is called a melting point. A melting temperature of DNA. Does that make sense to you? Now this transition looks just like the transition for actual like ice melting and stuff, something like that. Okay, so you warm up DNA, nothing happens to a certain temperature. Then around that temperature, in a very, very narrow range, absorption, light absorption, increases, stays high, and if you will decide to cool down, so let's heat, and this is cool, okay, and then you cool down. It cools down, the strands go back together. So far makes sense? Now, okay, imagine you have two DNA samples, two different DNAs. You can determine melting point for each of them. You know nothing about those DNAs so far, okay? So the first thing you do, you stick those samples in the spectrophotometer, the machine, and you measure the absorption of light. And you figure out that sample number 1 has melting point of 57. And sample number 2 has melting point of 65. What conclusion can you draw based on this? You know only melting points, yes? You don't know if they are. If I would tell you they are of the equal size, then absolutely. And then your conclusion is right. If we don't know the size, all we can say 65 is more stable than 57. That makes sense? That's it. If we know the length, then yes, we can. Now look, Chris made a great point. We can actually approximately see if those samples are of the same size. And if they are, then we can say there is likely more JCs in the sample with a higher melting point. Does that make sense? So it's actually an important parameter, and it's fairly easy to measure. Okay. Now, DNA is a pretty tough molecule. It can withstand heat. DNA molecule doesn't really care about freezing. You can freeze the heck out of it. Nothing's going to happen to it. Boiling can hold boiling for several minutes. DNA is pretty stable, okay, to environmental impacts. Unlike DNA, though, bless you, RNA, we had a joke that when you isolate RNA from the sample, if you look at it wrong way, it starts to degrade. I mean, it's a nightmare. Working with RNA is a nightmare. In some labs, what I saw, they have a dedicated area to work with RNA. It is squeaky clean. They're special because there are enzymes called RNase that breaks down RNA all over the place. And God forbid contaminate something with that enzyme. So those places are squeaky clean. You wear gloves. Sometimes it's a hood. So there's the airflow that prevents you from contaminating the place. They have special plastic wear, special set of pipettes, everything special. When you isolate RNA, you keep it on ice for a short period of time because then you want to freeze it at -80 and never throw it back unless you really, really need to. It's really, and it degrades anyway. It starts to decompose the minute you isolated it. It's very unstable. Well, sorry, I missed the time. So we're going to talk about RNA structure and why it's so unstable on Thursday.